question, or somebody asked why I was dressed up. And I'm dressed up because I'm trying to look as much like the picture that Sandra from Ireland drew, claiming that was me. And whether it looks like me or not, I leave you to judge. Now, I'm going to start by giving a recap from stuff I've done in earlier webinars. I'm going to talk about the stuff from them that you need to know for this one, so that people who are coming in for the first time don't miss out on the background information. And the first thing to say is that Cavernoma are formed on blood capillaries. And here we have a picture of a blood capillary in cross section. It has a lot of cells on the cell wall, a sheath around it. There are other cells that I'm not showing here. And Cavernoma grow from blood capillaries in some way. If you are one of the 99.9% of the population who don't have Cavernoma and don't get Cavernoma, then the contacts between these cells are what are called tight junctions, as you can see over on the left here. And these keep the cells tightly together so that red blood cells and indeed many other things can't get through the cell wall. That gives you a sort of a form of security, if you like. But if you're one of the 0.1% with a Cavernoma, where you have a Cavernoma, you've lost your tight junctions. And if there's somebody on, if we could mute, please, everybody, so we don't get extraneous sounds. Unless you want to ask a question, that would be helpful. So you lose the tight junctions and a Cavernoma can form. So this is a single cell that is, as it were, able to be part of a Cavernoma. So I'm going to be talking about tight junctions, and I use the abbreviation TJ for that. And the T, we talk about the genes inside these cells. Those are the tight junction form of the Cavernoma genes. And then over here, we have the CCM, I'm going to call them, type, a part of the Cavernoma genes. The next thing to say is that if you are one of the 99.9% with in the normal population, you have two chromosomes. Well, you have 23 chromosomes, each in pairs, one from your mother, one from your father, and on those chromosomes are one of three Cavernoma genes, which here are shown in their tight junction form. And if you have those, you have a normal blood capillary, no Cavernoma. But quite a lot of people, well, in the region of sort of the 0.1% or so of people are born with a familial Cavernoma gene, and that comes from either their mother or their father. So you would inherit a Cavernoma gene here, and you'll have a tight junction gene from your other parent. And that isn't sufficient to cause a Cavernoma. That is, that will still be a normal capillary. In order to get a Cavernoma, you have to have both of these genes in the CCM form. So you're born without that, either way, with both of them not, or with one of them not, and you have to develop that before you can develop a Cavernoma. And the way that happens is through what's called a somatic mutation. And a somatic mutation is one that happens during your lifetime. You have, I don't know how many billion cells in your body. There are many somatic mutations going on the whole time, but the chances of hitting this particular cell are fairly low. But when that happens, the tight junction form will be converted into the Cavernoma form, the CCM form, and then a Cavernoma can develop. So if you have the normal, both tight junction forms, you have to have what are called two hits in order to get a Cavernoma. And then what you get is what's called a sporadic Cavernoma. If you have the familial genes, it's a familial Cavernoma. So that's the background you need to understand for this talk. And Tracy, are you able to turn off people's mute for the moment or not? So that's what we have to do to understand today's talk. And today's question is the following. Everything I've said in previous webinars has concerned what happens in a single cell in a single blood capillary. It's talked about the changes that take place in this one cell. And I've talked about that in all the previous webinars. And so here we have a cell that's been talked about. But the question is, how do you get from that single cell to having a Cavernoma with this is what we're talking about. What is the development between this on the left and this on the right? And that picture is rather misleading because the real rough Richter scale is a much smaller blood capillary with cells here. compared to the Cavernoma out here. Of course, Cavernoma changed in size quite a lot, so you might say, what size Cavernoma is this? I'm not getting into that too detailed, but if you see a cell here, quite small, each of these lobes of the Cavernoma are composed of many cells. So in this Cavernoma with many lobes, it's quite a mature Cavernoma. This was one that was excised by a neurosurgeon from someone, I don't have a clue who. There are many cells in that Cavernoma. So what I'm going to do is give you the answer first, and that's fairly quick. And then what I want to do is talk about the science that produced the answer. If you're a scientist, you're going to have to do the research first and work out what the answer is. We're going to cheat because it's easier to look at the answer and then see what part of the evidence produces that answer. And so here is the answer. The evidence is from research on mice. You can't do these experiments on humans. And so is what's happening in mice the same as what's true in humans? Technically, we don't know at the moment, but what has been done on mice has been found to be the case in humans pretty much down the line so far. So the expectancy is this is what happens in humans as well. So we start off with a blood capillary. Here it is going left to right with the cells of the cell wall. And one of them gets a hit from radiation or whatever causes that initial hit to get that happening. And what happens then is you get a conversion to two CCM genes, whereas you only had one or two or none before, and that's the initiation of a Cavernoma. And step one is the first step. There are two steps here that happens in the development of Cavernoma. The first thing is that you get what's called a clonal expansion of this cell. So these cells here are identical to the cell that was hit initially. So they all have the same CCM gene in them. They are identical. And here they are colored red for this example. What happens next in step two? So this is a young Cavernoma. You wouldn't see this on, if you took an MRI of yourself, you would never see this. And what happens next is that you keep the cells that were the original clones, which you also incorporate normal cells here. And so here you have a slightly older Cavernoma, which has got three, what I might call developing nodes or lobes. And here, this one is formed from normal cells which have been incorporated. There's a slight slant on what a normal cell is here, which I'm not going to cover unless you ask questions about it later. So those are the two steps that you have in the development of a Cavernoma. And they then grow from there in the same way. You incorporate more cells. I'm not sure whether you get further clonal expansion or not. I suspect you do. Now, this was some research done by a group from Italy, France, and Sweden, and maybe other countries. Forgive me, I don't remember exactly where everybody comes from. But at the same time as that research was being done, which was 2 or 3 years ago, a similar research was being done in America, and their diagram from their paper is the one shown here. And it's basically the same. It's rather nice when two different labs get the same results, because that doesn't always happen. So here we have a normal blood capillary. The initiating step triggers this one. It's colored red again. And here we get proliferation of these cells. So they're all the same, what we've called a clonal expansion up top, but it's the same event. And then we incorporate cells from elsewhere, from other parts of the blood capillary. And that's what we call incorporate normal cells, incorporate neighboring cells here. So you can see the way that a Cavernoma is going to develop. And with further growth, further incorporation, you can get the complete Cavernoma. Now that's the answer. What I'm now going to do is talk about what the research was. And I'm going to present the research in a slightly simplified way because the advances in technology to do it have only been taking place over the last few years, some of them, and it's fairly intricate. So this is what I might call a slightly simplified view, but I can ask questions with a bit of luck, as long as they're simple ones, later on about the specifics. So the next slide is as follows. That is, well, how do we know? And the answer is these are going to be done on mice. And in order to do them, you have to have a breed of mice which has got a Cavernoma gene. And the scientific community has developed quite a lot of breeds of mice for research in neuroscience, not just on Cavernoma. There's something like 1,000 different lines of mice that are ready for people to do work on. So these are mice that have got familial CCM3. And because of that, they've got one CCM gene, a CCM3 gene, and one tight junction gene. And before they can develop a Cavernoma, they have to be hit in the normal course of events by radiation or chemicals or whatever will cause it, just as it would be in humans. So that's the starting point, familial Cavernoma mice. Now, what they have to have is a second hit. But whereas in life you get the hit from the radiation or whatever, in these experiments you're going to put these CCM genes in, what are known as transgenes. You may have come across transgenes, transferred genes into a cell. And so here we have what is going to be a transgene. It hasn't got into the cell yet, but this is a CCM gene. It's basically the same. It'll be a CCM3 gene for these experiments. And this particular transgene has been made in the laboratory by the research lab, and they've made it in a special way because they've incorporated a fluorescent dye on it. Here's a red dye. And so this gene is going to be inserted in place of a tight junction gene in the living mouse. And the way that happens is, you've seen this diagram before. This is a diagram I've drawn frequently with a tight junction gene and a CCM gene. And I'm going to turn it round so that it's that way round because that helps for the moment. And so here we have a tight junction gene inside this chromosome, this TJ gene. I've drawn out long. This is a long length of chromosome now, just with a tight junction gene in. And here we have the gene that's outside at the moment that's going to be inserted. And the way it's inserted, I'm not going to go into too much. It's fairly intricate, and it's gene technology, which I'm sure you've heard of. But you do a swap of the TJ gene for the Cavernoma gene down here, the CCM3 gene. This can get inserted into this cell and take over the TJ gene. And the way it can be done is dead cunning, because what you do is you have your CCM mouse, that's the bit up here with the TJ gene, and you then inject into the mouse these CCM3 genes, but they don't go in because this particular way of doing it has to have a trigger. And the trigger is tamoxifen. You've heard of tamoxifen in other ways, but here it's a trigger for being able to do this swap. And so you can inject the gene into the mouse and it just stays there, but only when you inject the tamoxifen will it cause this swap to take place. So we're going to use not just a red dye, we're going to be using 4 different fluorescent dyes called yellow, green, and it's actually cyan, but I'm calling it blue. It'll have a C mark and B marker for blue sometimes. So here we've got a whole mixture of 4 different kinds of CCM3 gene, differing only in the type of fluorescent attachment they've got to them. And any one TJ gene will only take one of these. So this cell might take the red one, but a cell over here will take the yellow one, and so on. And so you have, in fact, 4 different types, if you like, of cell, some with red dye, some with yellow dye, green and blue. One of these, CCM, comes from the origin. This is the transgene that's gone in. So different cells have got different types, different colors, if you like, of fluorophore, but they all have CCM3. So here's our green cell from the previous slide, and it's got injected into it a transgene with a green fluorophore, a green fluorescent dye. The other cells around here have got the same TJ CCM mix. So only this cell has got the transgene in it. But of course, other versions of the blood capillary, other blood capillaries have got a yellow dye. Here's one with the yellow, blue and red. These are all different blood capillaries. I can't remember how many miles, kilometers for the Europeans of blood capillary there are in a mouse, but in the hundreds. It's a long, there's a lot of blood capillaries in the brain. They're small. I'm not sure I mentioned that. These are only about the same diameter as a red blood cell, slightly bigger, but not that much bigger, so they're tiny. And so this blood capillary might be somewhere totally different in the brain to this one. It probably will be. And you can determine what proportion, of these cells have got a dye by the concentration you inject of these cells here. So if you injected lots of this, a lot of these might get transgenes in them. But if you do it at a low enough density, not too many do, but enough, and you can do experiments to get the right number. So that's the way the experiments start. And so you've injected your mice with these transgenes, and the mice develop cavernoma. And they develop cavernoma, in this particular instance, mainly in the cerebellum. This is a picture of a mouse brain. Here are the lobes of the brain, the cerebral hemispheres. This is the cerebellum. We all have a cerebellum at the back of our brain. And this is the brainstem going down to the spinal cord. And the eyes will be connected in some way up here. And the cavernoma are these red dyed things here. They're actually, irrespective of which dye they're always red, because it's the blood rather than the dye, the fluorescent dye on it, because we're looking at these in normal light. And what the experimenters do now is take sections, as I've indicated here, through the brain. You may have at school done histology and taken sections of tissues of various kinds. Well, the way this is done is you get sections. These might be the sections you have, but we're going to do the observation of these with a special microscope. It's called a confocal microscope, and a confocal microscope looks at fluorescence. The light you shine on it is dark to the human eye, but it excites the fluorescence. So that's the reason we put in fluorescent dyes to mark where there was cavernoma genes in the brain of the mouse. And so another speciality of the confocal microscope is you don't actually need to cut sections like this and look at them all. You can have a pretty thick section, and it enables you to look at a thin part of that bit by bit, just by the way the microscope focuses. But the point is, you get lots of different sections. Here's my rather bad view of what a cavernoma looks like. It'd be quite tiny, of course, in this space. And you can then look at it in different depths, if you like, at different parts of it, using your confocal microscope, looking down here. I hope you're okay so far. Not too many questions. What I'm going to do now is show some of the results that
David, excuse me. Before you go to results, we had a question from back to clarify.
Yep.
You may want to wait till the end, but someone asked, when you were talking about the hit that's needed to change that mutates the tight junction to the Cavernoma CCM, does that mean that in humans, let me get this right, that a cavernoma can be caused by radiation?
Yes, but I'm not talking about radiation like you might go to a radiologist for. I'm talking about the background radiation. We live in an environment where there's background radiation, not because of, particularly because of the environment we create, but there is radiation coming in from outer space the whole time. We might, you will also be taking in chemicals of various kinds. There's lots of reasons why that the hit takes place, but the radiation can be, if you have radiation, as it were, when you're very young for some reason. For example, you might have an X-ray done of a brain, then that can trigger this later on. That can be the initial trigger. So radiation that's applied artificially through X-rays or other ways can and undoubtedly does cause cavernoma from time to time, but you don't have to have that for the radiation that's needed here.
Okay, thank you.
So what I'm going to do now is to show some of the images that have come out of this research. These come from the Italian, Swedish, French group. And what you see here are two images, and the image is of a blood capillary which comes down at the bottom here and comes out at the top. There's another one over here. And the difference between these two images is that this one is done with a dye called podocolexin, and that stains all blood cells, not just the ones that have got CCM genes. Whereas the cyan, green, yellow, red are shown here, but they're also shown over here. So all you see here is the cells that are CCM3, in this case, origin. And almost all the cells here are, of course, and most of them are red. And the density of the cells, there's a lot of cells here, because this is a very newly forming cavernoma at this point here. And only one, or at the very most two of these cells, there are many thousands of cells here, will be the initiation one. Almost all of these are the clonal expansion. And we can make it rotate so you can get some sort of idea about what it looks like. And so you can see that up here there has been a blue source of CCM3 and a green one, but the bulk of these are the clonal expansion of a newly developing cavernoma. Here's the blood vessel coming in, coming out again, and there's another one down here. So that is this particular early stage cavernoma. But we can stop that, and we can go to another stage. Here is another stage where we've got a blood vessel, a blood capillary coming down here. You can see because it's white, but it isn't shown up here. And here we've got a first lobe of a cavernoma, which is primarily red, but with gaps in. So this is, if we make this rotate, you can get a sort of image about what that's like. And so here we have what I would call a step 2 image. This lobe here is mainly red, but there are gaps, and the gaps aren't showing up white here, but they would be in other ones. I don't have an example of that. And this is where you're starting to incorporate other cells. This is like a balloon with some CCM3 cells in various parts of it and some tight junction in other part of it. And so that's of step 2 stage cavernoma. But if we take another example, I can stop, I don't need to stop it, I don't suppose. This is at the stage where it's a raspberry, and it's a bit complicated to see. And there are some quite nice diagrams down below which can help. And I'm going to rotate it first of all, and then I'll talk through one or two of the aspects of it which are important. So here we have a fairly thick section now, done by this confocal microscope, and you can see the three-dimensionality of it quite well because of the rotation that you have here. And I'm going to stop that and start it again at a particular stage. Well, it's not a bad stage here. On here at the left, this shows you the lobes of the cavernoma. So there's one up here, which is mainly green stain, we'll see. Here's another lobe over here, which is mainly yellow. Another lobe we'll find somewhere down here. This is an early stage cavernoma. It doesn't look quite as neat as you get in the drawings, but this is the research that led to those drawings. So we'll rotate it again, and you can see this lobe up here I want to talk about. Let's stop the trace here, because here we have a lobe of the cavernoma, and it's now part of a raspberry, and it's got a number of green dots here with a lot of blank space, which is white over here. These green dots have CCM3 genes in them. These are cells with CCM3 genes, not single cells. These would be clusters of CCM3 genes. And here we have ones that are tight junction genes. These are the ones that are incorporated into the cavernoma.
David, can I stop you? While you're stopped there on that, we have another question to clarify that leads to this. Were all the dyes injected into the same mouse at the same time, all those 4 dyes?
Yes, they were. This is a mouse that had equal concentrations of all 4 dyes injected into it at a level that enabled you to not get the whole thing plastered with the injections, but on the other hand, enough to get this result. These results, all 3 of these traces are from a single mouse. The paper dealt with a lot of mice because the research wasn't only done on a single mouse, but these experiments, these 3 ones I've shown you, all come from a single mouse. So, so this is really the end of the story of the slides I want to show. And so we'll go back to the forms. I really meant to show while I was doing this, excuse me, these, because what we have in here is a beyond stage 2. What we had in this one was a stage, was a stage 2. And what we had in that one was a stage step 1 cavernoma, embryonic cavernoma. So that's the evidence which allowed you to conclude these results. And whether you find that exciting or not, I don't know. I was thrilled when I saw these papers because I had no idea. 3 years ago, I wrote around to people who I thought might know, what is a cavernoma? What are the lobes? Do they come from a single capillary? Are they formed from groups, from lots of capillaries and so on? And this gives the answer. It's totally brilliant. So, what use is it? And this isn't for this talk. I'm doing another talk later on about the sort of drugs that are being developed. And my science talks so far have all been basic science, I would call it. They've all been simply in order to do an understanding of cavernoma. But always at the back of one's mind is, before you can get the major advances, you have to do the basic science. Here, there are 3 places where you might expect to get drugs working to stop cavernoma forming. Either at the targeting of the TJ junction, so you don't get conversion to cavernoma, to the CCM gene, excuse me. That's one source, and that's actually being done. And there are drugs in development in clinical trials at the moment which are working on this level. You might try and prevent clonal proliferation. I didn't give you the full story about clonal proliferation. It's slightly more complex than I said, and I can answer a bit about that if you want. But you could prevent the clonal proliferation. That would stop this stage getting any further and prevention of the cavernoma. Or you might stop the incorporation of normal cells here. And this research is being done not only at the level of the research I've shown you today, it's also being done at looking at the biochemistry that's taking place in all of these. And CA UK has a strategy document, and its title is Cavernoma 2030. And what it's saying is that we hope that by 2030, we will be able to prevent cavernoma forming if they form. And it's this kind of research that leads to that. So if I can just give a summary of what this talk's been about. First of all, you have capillary blood cells in the normal 99.9% of the people that have tight junction genes only. If you have the familial form, you have one chromosome with a tight junction gene and one with a CCM gene. You then have to have a hit. So a somatic mutation is that hit. In the life, you have a somatic mutation that takes place, as I've described, during your lifetime. If you're doing these experiments, the equivalent of the somatic mutation is the injection of a transgene. That is the equivalent of the hit. That cell is then cloned many times to form the early stage cavernoma, each with 2 cavernoma CCM genes. And that early stage, step 2, incorporates cells from elsewhere that have tight junction genes, but still get incorporated into that cavernoma. It's slightly more complicated than that, but that was the explanation to enable you to get the essence of it.